Hi, in this video we'll be talking about cellular respiration and photosynthesis. Cellular respiration is one of the major sets of catabolic reactions in a cell. During cellular respiration, fuel molecules are catabolized into smaller units to release the energy stored in their chemical bonds so the cell can do work. Cellular respiration takes place in 4 stages. The 4 stages consist of glycolysis, acetyl-CoA synthesis, the citric acid cycle, and oxidative phosphorylation. Stage 1 of cellular respiration, glycolysis, is a set of 10 chemical reactions broken down into 3 different phases. These phases consist of the preparatory phase, the cleavage phase, and the payoff phase. In the preparatory phase, glycolysis starts off with one 6-carbon molecule of glucose and eventually ends up with 2 3-carbon molecules of pyruvate. In this phase, the addition of 2 phosphate groups is required to convert glucose into fructose 1,6-bisphosphate. The energy required in order for this process to occur comes in the form of 2 molecules of ATP. During the cleavage phase, fructose 1,6-bisphosphate is broken down into 2 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which is then converted to a second molecule of glyceraldehyde 3-phosphate. In the final stage of glycolysis, the payoff stage, the resulting products consist of 2 pyruvate molecules and 2 NADH electron carrier molecules. Additionally, 4 ATP molecules are produced, but since 2 ATP molecules were previously used in the first stage of glycolysis, the net production of ATP from a single glucose molecule would be 2 ATP. After glycolysis, the next few stages of cellular respiration take place inside of the mitochondria. Mitochondria have 2 membranes that define different spaces. Between the inner and outer membrane, there is the inner membrane space, and inside of the inner membrane is the mitochondrial matrix. In aerobic reactions, pyruvate is broken down even further to release more energy. It is first converted into acetyl-CoA and then goes through the citric acid cycle. The breaking down of the pyruvate from glycolysis takes place inside of the mitochondrial matrix. First, the pyruvate is broken into CO2 and an acetyl group. Coenzyme A transfers the acetyl group to the citric acid cycle. All of these molecules are catalyzed by enzymes called the pyruvate dehydrogenase complex. This reaction produces 1 CO2 molecule, 1 NADH molecule, and 1 molecule of acetyl-CoA. Since these were also produced during glycolysis, after this reaction, we have 2 of each molecule. Now, here's a short video clip explaining what goes on during the reaction in phase 3 of cellular respiration, the citric acid cycle.
1 molecule of acetyl-CoA enters the citric acid cycle. The 2-carbon fragment of acetyl-CoA attaches to the 4-carbon molecule, oxaloacetate, in the first reaction of the cycle. This forms citrate. In a series of steps, bonds break and reform. 2 carbon atoms are released one at a time in molecules of carbon dioxide. Electrons are carried off by molecules of NADH and FADH2. One step produces an ATP molecule by substrate-level phosphorylation. A 4-carbon oxaloacetate molecule is regenerated.
Now, onto the 4th and final stage of cellular respiration, the electron transport chain. The electron transport chain is located in the mitochondrial inner membrane. In this stage, NADH and FADH2 push out protons and transfer electrons. In complex 1, NADH pushes out a proton or hydrogen atom and becomes NAD+. Also, it transfers an electron to complex 2 with the help of coenzyme Q. In complex 2, FADH2 becomes FAD while pushing out a hydrogen proton and transferring an additional electron. The electrons travel through complex 3 where yet another hydrogen proton is pumped to complex 4 with the help of cytochrome C. In complex 4, oxygen attains the electrons as well as a few hydrogen atoms to produce water. The remaining hydrogen atoms remain in a state of high concentration in the intermembrane space. The protons need to be transferred to an area with low concentration like the mitochondrial matrix, but can't without a transmembrane protein channel. This intermembrane protein channel is created by ATP synthase. ATP synthase has 2 subunits, F0 and F1. The proteins flow through F0 into F1 where ATP is synthesized. A cell's ATP level is the amount of energy it has at a given time. The goal of cellular respiration is to produce as much ATP as possible. Whenever the ATP levels are high, the generating processes are slowed down. However, whenever the ATP levels are low, the cell activates the pathways that lead to ATP synthesis. Now that we've finished talking about cellular respiration, let's move on to photosynthesis. Photosynthesis is a redox reaction. It is the process of converting carbon dioxide and water into glucose and oxygen. The oxidation of water in this reaction is associated with the reduction of carbon dioxide through a series of redox reactions which creates a photosynthetic electron transfer chain. Photosynthesis starts by absorbing light using protein pigment complexes called photosystems. Photosystems use this absorbed light energy to fuel redox reactions and set the electron transport chain in motion. The electron movement drives the synthesis of ATP and NADPH. In plants, the main organelle are chloroplasts. Chloroplasts contain chlorophyll, which is what gives the plants their green color. Chloroplasts contain of 6 parts: the outer membrane, inner membrane, thylakoid membrane, grana, lumen, and stroma. The photosynthetic electron transport chain resides in the thylakoid membrane. Along with photosynthetic electron chain, the Calvin cycle is another major part of photosynthesis. The Calvin cycle consists of 15 chemical reactions to synthesize carbohydrates from CO2. The 3 stages of the Calvin cycle are carboxylation, reduction, and regeneration. During carboxylation, CO2 is absorbed from the air and added to the 5-carbon molecule ribulose 1,5-bisphosphate. The enzyme that catalyzes this reaction is called rubisco. After the reaction is complete, the resulting 6-carbon molecule is broken down into 2 3-carbon molecules of 3-phosphoglycerate. During reduction, NADPH is used as a reducing agent to reduce 3-PGA. This takes place in 2 steps. In the first step, ATP is used to phosphorylate 3-PGA, and then NADPH transfers 2 electrons to the phosphorylated compound. This results in the formation of a triphosphate, which are exported from the chloroplast. Finally, during regeneration, the 5-carbon molecule used in carboxylation is regenerated. This phase counts for the majority of the Calvin cycle. Overall, 3 NADPH and 3 ATP are needed for each molecule of CO2. Here's a short clip that briefly describes the Calvin cycle.
During the Calvin cycle, carbon dioxide, CO2, is added to a 5-carbon molecule called RUBP. The resulting 6-carbon molecule is unstable and quickly splits into 2 3-carbon molecules called 3-phosphoglycerate. Using energy from ATP and reducing power from NADPH, which are products of the light reactions, the pair of 3-phosphoglycerates move through a series of reactions and are converted into 2 molecules of glyceraldehyde 3-phosphate. When several of these glyceraldehyde 3-phosphate molecules have been produced, some combine to make glucose, while others are reused in the Calvin cycle. To generate an entire new glucose molecule, the cycle has to turn several times, because each turn of the cycle adds only 1 carbon atom from each molecule of carbon dioxide.
Photosystems are critical because they convert absorbed light energy into the movement of electrons. The photosynthetic electron transport chain connects 2 photosystems. Both systems are necessary in order to pull the electrons from water and use them to reduce NADP+. Photosystem 2 produces the electrons for the beginning of the electron transport chain. After 1 electron is lost, another is pulled from water. The electrons are energized with the second light input supplied by photosystem 1 in order for them to have enough energy to reduce NADP+. When passing through photosystems 1 and 2, the electrons also pass through the cytochrome B6F complex. Plastoquinone carries the electrons from photosystem 2 to the cytochrome B6F complex, and plastocyanin carries the electrons from cytochrome B6F complex to photosystem 1. Because the level of light intensity changes throughout the day, the photosystems have to work in a cyclical manner. When the levels of light are low, light is evenly shared between the 2 photosystems. Although when the levels of light increase, the amount of light energy absorbed starts to overwhelm the ability for the Calvin cycle to make NADPH. When no NADP+ is being returned, this can damage the cell. In order to stop this from happening, electrons are guided into a different pathway that leads to an increase in ATP production and a decrease in NADPH production. One interesting fact that you may not know is that cellular respiration and photosynthesis are like opposite reactions. The that are created in photosynthesis are actually the reactants used in cellular respiration. Hopefully, after watching this video, your interest in photosynthesis and cellular respiration and all other chemical reactions has greatly increased and you continue to do research.